This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 631, recorded on June 24, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Madison, New Jersey, Brienne Barker.
Hi, it's great to be here. It is beautiful and sunny and 82 Fahrenheit here.
I can't really see. I'm in the basement. I'm looking out the window. I see some sun, so I'll take your word for it. Also joining us from Austin, Texas, Rich Condit.
Hi there, Vincent. Hi, Brienne. It's 81 degrees, mostly cloudy. It's only going to go up to 86 today. We've had a little rain. We're going to have some rain later in the week. It's fine. It's okay.
We're only going to go up to 86.
Other things are pressing, aren't they?
Say what?
Other things are pressing.
Yeah, there's a lot of coronavirus out there.
Going up, huge. Big time in the U.S., going up.
Yeah, and Texas is... not necessarily leading the pack, but it's on a run. Yep. A couple of days ago, we had our peak at 500 cases in a day, and it's really spiking.
Yeah.
And I know that they're starting to get a little nervous about the possibility of the healthcare system becoming overwhelmed. I mean, they're not flipping out yet, but they are making contingency plans in Austin for turning a convention center into a an overflow hospital. So they're trying to prepare themselves for the worst and trying to encourage people to behave themselves. But from what I can tell, there's a lot of what I would call misbehavior out there.
Oh yeah, big time. This is all because people are going back to their normal lives and not doing it right. And it's partially their fault, but it's also partially someone else. Their fault because they're not all wearing face masks. I saw a picture of Lady Bird Lake in Austin.
Oh.
Yesterday in the Times.
Yes. As a matter of fact, that wasn't actually, this is great, that wasn't actually Lady Bird Lake. That was Barton Creek, upstream from Lady Bird Lake, which I know well because I go kayaking there with my daughter and my grandchildren. We were, as things were starting to open up and before things started to really spike, I was talking about, maybe we ought to go kayaking again. That's pretty distant. And that picture is almost exactly where we usually beach our kayaks, to swim in the river and stuff. And it was packed. I'm not going.
Yeah, well,
I think that's probably a good call.
They didn't have any face masks on, and I still see a lot of people around without face masks. That's part of it. But the other part is we should have been testing more. I think everyone should be tested once a week with some kind of rapid PCR that should have been developed years ago, and the government should pay for it. And that's all a pipe dream, I know, but that's what the shutdown was for, to get ready for that. And we didn't do it. So, folks, if you're pissed, you should be, because we're still caught flat-footed. And that, people ask me, why did we shut down if it was going to start again? We shut down to be ready, and we didn't do anything because there's no guidance. And so that's my feeling for today. And yesterday and tomorrow. I wanted to... I just got an email about an hour ago from Human Vaccines Project. I don't know what this is, but it's an interview with Paul Offit. And I made a PDF out of it. I'll put it in the show notes. He's talking about vaccines, really good thoughts, because he made the rotavirus vaccine. He was a developer of it. And he's talking about, vaccine development, what's going on, how's it going to be tested? And he says some cool things. Let me just point out here. He said, he was asked, is there a potential trade-off between speed and safety efficacy in the race to develop vaccines? He says, I'll tell you in 8 months, the phase 3 trials will tell, assuming that we do phase 3 like we've been asking them to do, which involve at least 20,000 vaccine recipients, 10,000 placebo. If at least we do that, we'll see. So, I mean, he's saying this is what we should do, but who knows if we actually...
And he said 8 months?
8 months.
to conduct that trial.
Yeah. And it's not phase 3. Nobody's in a phase 3 yet, right?
Right.
And then here's the best part. What keeps you up at night? It's a Rich Condit question. He says, there is a system which I trust that has been in place since the 1950s to make sure that the vaccines that are brought into this country are tested as much as is reasonable to mitigate risks regarding safety and efficacy. This system involves the NIH, the CDC, and the FDA. As long as that system stays in place, I'm good. What worries me is that this system could be perturbed by an administration that perturbs the science. This is an administration that took the words climate change off the EPA's website. This is an administration that pushed hydroxychloroquine and got the FDA to approve it, a product that had never been shown to work, was known to have a certain level of toxicity, and ended up doing more harm than good. That was the FDA at its worst. They let themselves be pushed around, and if it happens here, that would be a problem. Isn't that great?
That is fantastic.
Because I just did a vaccine lecture in class today, and one young lady asked me, are you worried about any aspect of the testing? And I couldn't put it as good as he did. So I just sent this to everybody in the class because it's true. Come on, keep the politics out of science. You guys, you don't know how many letters I get yelling at me, telling me to keep the politics out of TWiV. And you know what? I can't if they're going to mess with us.
Yeah. When the politics messes with the science, then you gotta push back.
They let us make the vaccine the way it should be made, the way he describes there, then we're all good. But if not, then we have to talk about the politics and why the politics is getting in the way.
So we mentioned phase 3 trials. In fact, according to the draft landscape of COVID-19 candidate vaccines posted by the WHO, as of June 24th, 2020, there was in fact one vaccine that is listed as phase 3 trials, and that is the Oxford vaccine, the chimp adeno vector vaccine. Now, I don't know whether that's just that they've been approved for phase 3 or they've actually started recruiting or whatever. I'd have to look. There's a link here, but I haven't had time to look at it.
Yeah, I think they've been approved maybe, and they are going to...
Says overall trial status ongoing, recruitment status, recruiting.
Yeah, recruiting. Okay. So let me show you a cool figure that I got from the Millikan report, which tracks vaccine. Can you see? Yeah, you can see that, right? Look at this. These are all the vaccines, 172.
Wait a minute. Am I?
You can see my screen, right?
I can see your screen, and this is actually also the thing that you posted on social media earlier. Everywhere. Which I pulled up while Rich was talking. It was like we were thinking the same thing to look at this graphic. Yeah, because there's no phase 3, but maybe unless you're actually doing it. Anyway, Rich, can you see it?
Yeah, I can see it now, yeah.
So this is brilliant because it's categorized by type, and then on the top, then each line is a different vaccine, and then you got phase 1, 2, 3. And you can see most of them aren't even in phase 1.
Right.
And most of them are protein subunit based, right? That's the orange. And a couple are in phase 2 and a couple are in phase 1. So basically, this is going to take a while, folks. And you know what? If one vaccine gets through soon, it's not going to be enough for everybody. So we need more than one. Anyway, this is by the Millikan report. I just thought it was great.
Yeah. Are you going to put that link in the show notes or something?
I will.
So for some reason, I thought that the Oxford vaccine was a replicating viral vector, but looking at this, it probably is a non-replicating.
That's correct.
Yep.
It's a standard adenovirus with E1 region knocked out and a bit of the E3. They've tinkered with E3 and E4. So all of the basic regulatory stuff that would launch a replication cycle is gone.
Got it.
So it just goes in and expresses the transgene.
All right. I got 2 brevias for us to chat about.
Heavy duty brevias.
They're both really interesting.
And one of them, Brianne will help explain to us. because it's got immunology in it.
Okay, I can do that.
Let's do the first one is intrafamilial exposure to SARS-CoV-2 induces cellular immune response without seroconversion. Intrafamilial, that is, you're in the same family.
Yes. So these are within sort of household groups.
Right. So this is from Strasbourg University Hospital, Strasbourg, France, and the University of Rouen in France. Gallet, Villay, Wendling, Nazon, Partisani, Siblia, Candon, and Fafi-Kremer. And it's a med-archive preprint. Are we ever going to see papers again? Who knows? Who's going to review all this stuff?
Not me.
Anyway, so
that's a good question.
The question here is antibody and cellular responses. What's going on? And they use family to get at this, right?
7 households.
So they have 7 families, right? They have 9 index patients and 8 contacts. And they measure antibodies and T cells, right, from these patients. And you can tell us how they do the T cell assays. Yes. But basically, they're measuring antibodies to the virus and a T cell response. All the index patients recovered, so they had mild infection. They all developed antibodies. They had a good T cell response, which lasted up to 69 days.
Yes.
6 of the 8 contacts had symptoms within 1 to 7 days after the index patients, but they were all seronegative. 6
So wait a minute. When you say 8 contacts, that's 8 total contacts?
Yeah.
So I'm sorry.
Do you have the paper up?
Yeah, I have the paper up.
If you scroll to the end, there's a table 1, that shows all of the members of all of the households.
Okay, household 1.
Looks like it has parent 1 and child 1.
So I see. So in many cases, we're talking about just 1 case and 1 contact. And in, so a P is a patient and a C is a contact. I see.
Okay. Yep.
And so in household 3, there are actually 3 patients and 1 contact. Household 4, there's 1 patient and 2 contacts. All the rest, there's 1 patient, 1 contact. Got it. Very good. So
some of the point here is that 6 of the 8 contacts had symptoms, seronegative, but they had T cell responses against different structural and other viral proteins.
So all of the patients were symptomatic. Is that correct?
All the index patients were symptomatic. That's right.
And they all had an antibody response?
Yes, they were all positive based on serology.
Yep.
Were they identified originally because they had symptoms in a positive test? Or something like that? Because I'm thinking about this next paper we do that really addresses asymptomatic infections a little more strongly. It doesn't really matter. These were all people who were symptomatic, seroconverted ultimately, and you're looking at what happened to their contacts.
So they had, each of these households had at least 1 index patient with a documented PCR positive and or serology testing. Okay, so they had these, they were definitely SARS-CoV-2 positive.
And then it says all of the index patients recovered from a mild form.
Yes.
In March.
Right.
So they were all infected. And the reason this is interesting is because you got a family, you got people living together, and you can track the infection. You know there's someone infected there. You know there's contact with other people. So you can really time the infection to a window and see if other people get infected or not, right?
Right, exactly. And so I feel like I've said multiple times that I want to see T cell data. I want to see T cell data. I want to see T cell responses. And so this paper is nice in that they've got 2 figures looking at different T cell responses. They use something called an interferon gamma ELISpot. So they took peptides from the virus and incubated them with T cells that they had taken from these patients, and then looked at the ability of those T cells to produce cytokines in response to the peptide. And so specifically, they looked at the ability of those T cells to produce interferon gamma, which is an important T cell cytokine. And so they're measuring the ability of those T cells to make cytokine in response to a few different peptides here. And what's really interesting is that out of all of the individuals tested, both the patients and the contacts, only 2 were negative for T cell responses. Mm-hmm. All of the others were making some type of T cell response. And that is considerably more positives than they saw with their serology test. So there were quite a few individuals here who were negative with serology, but positive with T cell responses.
And of course,
am I reading this correctly that there were even quite a number of individuals that were negative for viral RNA in a PCR test?
Yes. So there are.
But I don't know when that was done. Do you know? And was that done in a fashion where they could have had an infection and cleared it?
My interpretation was that yes. The blood samples were all collected in May, and the index patients were sick in March.
Okay.
And so they could have spread to their contacts.
So they could have just done the whole thing and been done with it.
Yeah,
exactly.
Okay.
No, but if you're making T cells, you were probably infected. And some of the peptides they used were specific for SARS-CoV-2, right?
Yes, they were using peptides from SARS-CoV-2.
Because I know some of the like nuclear protein is the epitopes can be conserved among other coronas and cross-react, but at least some of the peptides they used were SARS-CoV-2 specific.
Yes, they were. And they looked at unexposed healthy donors as well as the contacts and the index patients. And there were some T cell responses to a few epitopes in the unexposed healthy donors. Maybe those are cross-reactive with other coronaviruses, but not nearly the level of response that they saw in the contacts or the index patients.
So I like this because people are doing serology saying, oh, there's no antibody response. This is horrible. The vaccines are not going to work. Well, there's T cell response. Maybe that's crucial, right?
Yeah, absolutely. Whenever I hear someone mention the lack of antibody responses, I think back to our conversation with Jonathan Udell, where he was wondering whether this virus was doing something to B cells and messing with B cell responses. Yeah. And every time I keep seeing data like this and thinking of that again and again.
Yep. Yeah, no, I think this is interesting, and certainly it should be expanded with many more people, right? To see, a lot of these studies with low antibody responses after recovery should look for some T cells, right?
Oh, yeah, absolutely. I think that trying to understand the T cell response is going to be incredibly important. And I remember that one of the other papers we talked about looked at whether there were cross-reactive T cells in responses from other coronaviruses. Yeah, and so understanding this T cell response may be a key part of understanding these immune responses.
So I'm thinking correlates of protection here, right? Mm-hmm. And we've talked about this before. And it could be that the T cell response is one of the major correlates of protection that we need to be looking at. These people got the disease, recovered, no antibody, got T cell responses. That says to me anyway, correct me if I'm wrong, that probably their recovery has to do with innate immunity and a T cell response.
I think it certainly supports that.
So now I start thinking about vaccines because a lot of the vaccines. Okay, first of all, do they comment on what peptides these people their T cells are responding to? Is it all spike? Is it other proteins? Do you know?
So it looks like there are quite a few to matrix. And I see some to spike, some to matrix, some to N, so the nuclear protein.
Okay, because
they have the figures with those epitopes broken, or the peptides broken down in the data.
Okay, because, I see. Most of almost all of the vaccines, and certainly I think, inactivated vaccines, that's going to be a whole virus, which is going to have all of those things in it.
Yep.
It's going to have spike, matrix, nucleocapsid, etc.
I think most of them, though, are spike only.
Yes.
Yeah, most of those proteins. And we saw in that previous figure that most of the vaccines that are out there are protein only.
Yeah, many of the all the vectored ones are just spike, right?
Yes.
Yeah.
That's right. And then you got to think about what sort of an immune response the various vaccines stimulate. Because if it's just protein, you're going to get mostly an antibody response, right?
Absolutely.
And that's where things like adjuvant come in. And other components of a vaccine to try and stimulate a T cell response as well. So not all these vaccines are going to work the same way. And
if at all, if at all.
Yeah. And if non-spike T cell responses are an important component of immunity, some of them are missing the mark.
Right.
Yeah, people have made a big assumption here going into these vaccines, right?
Absolutely. The one sort of caveat to all of this is that we don't know if the virus is messing with any types of immune responses. So if, for example, the virus was messing with B cells and antibodies, the vaccine might elicit antibodies really well when we don't have that inhibitory aspect.
Right. So that brings up another aspect of this whole thing. One approach, it seems to me, theoretically, to vaccines is to try and, as closely as possible, mimic the natural immune response. But that's not, is it not perfectly reasonable that you could stimulate something that is not exactly the natural immune response, but works just as well?
Yes. And there are a few vaccines that seem to really work that way. So if you look, for example, and I am drawing way back on things from graduate school. I remember
it's only last year, man. You're young.
Not quite. I remember we talked a lot about the importance of the CD8 T cell response in measles infection, and yet the measles vaccine drives a pretty large antibody response.
Oh,
this is just, if it weren't so deadly, this would be fascinating. Well, it is fascinating.
Yes.
But my fascination is tempered by the fact that people are dying.
Absolutely. And that measles information largely came in conversations with the person who is now Stephanie Langel's PI. So I'll have to double check it with her.
So this brings back so to summarize what we're saying is that T cells, people have T cell responses. It could be really important for recovery or protection or both. It could be that the virus is somehow messing with the antibody response and you get low antibody, but still T cells are there and that's helping. And if you make a vaccine where that messing protein is not present, which is probably all the spike vaccines are that might be okay because neutralizing antibodies are good. However, we did a paper on Friday on TWiV where they showed that they compared symptomatic and asymptomatically infected people. And the asymptomatically infected people had way lower IgG responses than the symptomatic people. Mm-hmm. That doesn't mean that the antibodies are making them sick, but it's a correlation, right?
Sure.
I'm wondering if you make higher antibodies, that's contributing to immunopathology, right?
Perhaps, or are you just making more antibodies because you have more virus?
Could be. Yeah, it could be either way. I agree. I would like to think that would be better, right?
Yes, you would hope so.
So anyway, it's fascinating.
The problem here is that we don't really have a lot of T cell rapid tests. It's not as easy to do a T cell assay.
Yeah, right.
As it is to do some of these antibody assays, and so this is more challenging to measure than some of the antibody responses are.
Totally.
There is no such thing as a rapid T cell test, right?
There isn't.
You got to culture cells.
You've got to culture cells. There are some things that you can do with flow cytometry that are a little shorter, but you need to have some pretty sophisticated flow cytometry.
Those are like tetramer. I'm really on the edge of my...
You could use tetramers are one way of doing it.
Okay.
The paper we did a couple of weeks ago from, I have his book here. What's his name? Shane Crotty. where they threw, hundreds of peptides at T cells just to see which. So that's a different way where you want to know what the repertoire of T cells are recognizing. Whereas here, you just take a couple, and the idea is just to stimulate the T cells that you suspect are there, right?
Right, exactly. Although if you look at, their figure 2, they are using peptides from different parts of the virus.
Yeah,
so
it looks like they got 9 different pools, I guess. I don't know if they're pools or what.
They're probably pools.
Couple of spike, as you say, nucleocapsid matrix, a couple other proteins. There are a lot of the T cells are seeing a lot of different epitopes.
Mm-hmm.
This makes me think of the dengue vaccine thing, where the dengue vaccine that's in the yellow fever vector has just the dengue envelope protein in it.
Yeah, it's right.
It had a number of problems. But suffice it to say that an engineered attenuated dengue strain that has a bunch of other authentic dengue proteins in it works better.
Yes, that's right. And that's what they think, that the T cells aren't being induced by the dengue in that yellow fever dengue hybrid. There's no T cells against dengue.
So in theory, at least, there's a lot, and with some support, there's a lot to be said for presenting a spectrum of antigens rather than just saying, spike is the whole story.
Absolutely. Because T cells, respond to a receptor that includes MHC, so that's a host gene that's different from person to person. So if you give a whole lot of peptides, there are more choices to bind to those unique MHCs.
Wow. We're throwing a lot into spike, man.
Yep. Well, I mean, I had the...
Lots of eggs in the spike basket.
That's a great title. Lots of eggs in the spike basket. People won't like it. They'll get mad at us.
So let me see. The Oxford vaccine is spike. The Ad5 is spike. mRNA vaccine is spike. I'm reading down the list of the guys in clinical trials. There are 3 inactivated vaccines, all from China. And those would then have a spectrum of...
That would be okay.
They would have a spectrum of... yeah.
There's also a couple of attenuateds that might be okay too, right? Or not. Maybe not.
There are no attenuated vaccines in trials. There's another spike, RNA. That's a protein subunit is spike, RNA is spike. Oh, here's another inactivated from another. It's also Chinese. So that's 4 of those. 2 DNA guys. Those are going to be spike, non-replicating vector, adenobase. I presume that's spike. 2 other proteins. Those are going to be spike. So there's of the, I don't know, it's 12 or 14 vaccines in clinical trials now. 4 of them are inactivated. That'll have a spectrum of antigens. The rest of them are spike.
I was thinking about this morning, and 172 vaccine candidates. So a lot of these companies have never made a vaccine ever. And so you can imagine they had a meeting. A couple of people sat down around a table. And they said, hey, we have this vector. Why don't we throw spike into it? Okay. And that's it. That's the meeting.
Right.
Yeah. Well, fortunately, it does look like spike leads to some pretty good T cell responses. So all is not lost.
I agree.
Well, I mean, a spike vaccine might work just fine. It may not, as we were saying earlier, it may not exactly mimic the natural immunity, but it may work just fine.
We're just playing with the data. We're being critical. It's okay. That's what we do.
I'm sitting here just thinking, I'm astonished at all of the science that's being thrown at this globally in such a short period of time. Has there ever been a disease that has been examined in this kind of detail by this many people in this short a period of time? I don't think so. It's amazing.
Certainly not this quickly.
Yeah. I mean, you saw a precursor of this with Zika, right? A lot of people jumped on the Zika wagon, but I don't think as many as this, because that never hit as many people, though.
But all of this is possible because we've been doing so much virology and immunology to set us up to do these things quickly.
Yeah. Yeah. Okay. The other brevia is it's actually an A-archive paper, which is like the original server for preprints, before bioRxiv, before medRxiv. It's called Probability of Symptoms and Critical Disease After SARS-CoV-2 Infection. This is out of Italy. The first author is Poletti, and the two last authors, Aielli and Merler, from Milano, Trento, Pavia, etc. So the question here is, as the title says, how many people have symptoms and how many of them are serious? And so they had a lot of people in Italy to do this study with, and they had this one study between February and April in Lombardy. They did contact tracing to investigate the history of exposure of 64,252 close contacts of 21,000 confirmed cases.
Amazing.
Amazing is right.
That's awesome.
A close contact is someone living in the same household or who engaged face-to-face within a distance of 2 meters for more than 15 minutes.
Like you would do at a bar. People.
Yeah, or you could, in the old days when we used to have meetings in science, we would do that.
Sure.
You could stand in front of people and talk for, or,
shout at them.
I mean,
pre-Zoom.
Face-to-face. I mean, have a committee meeting with your student across the desk, right? The chair is close. So anyway, that's what face-to-face means. And then they investigated these close contacts and looked at them for symptoms, basically, and RT-PCR.
Yeah,
right?
Yeah. They first collected who's PCR positive,
right?
And how many PCR positives did they come up with?
I think it's 5,484.
Something like that.
Yeah. So they had 5,484 close contacts positive, right?
Oh, wait a minute. The 5,400 were assayed.
Yeah, they were assayed. So of those, half of them, 50% were infected. Okay.
So 2,800 were infected.
2,800 were infected. Let's see here. The median age was 53, 56% female. One of those, 1,800 were serology only and 600 PCR only and 300 by both tests. So they did a variety of tests. And then they looked at symptoms in these, basically.
This blows my mind. This blows my mind.
So the probability
half of the contacts were infected. There's a number already.
That's right.
Right.
Okay. If you're that, just extrapolate that. If you're in contact with somebody and with that sort of contact with somebody who's infected, you got a 50-50 chance of getting infected. If I extrapolate that. So that's pretty heavy duty.
Symptomatic cases were defined as subjects showing upper or lower respiratory tract symptoms like cough, shortness of breath, or fever. Critical cases were if you went to a hospital or you died. So that was considered critical. All right. So the probability of developing symptoms increased with age. So 18% if you're less than 20 years of age, to if you're 80 years or more, you had a 65% chance of developing symptoms. People who did not show fever or respiratory symptoms were 74% of all infections younger than 60. That's one of the big takeaways from this study.
Do that again?
Yeah.
74% of all infections in people less than 60 were asymptomatic.
Right. Okay. So
74%. If you're younger than 60, you don't even know you're infected. And that's why this thing is spreading, right?
Right. Whoa.
All right, so that's one thing. And here's the other. Critical care was required for 0.54% of infections in people under 60. Over 60, 6.6% required critical care. I mean, that's going in the hospital or dying. So that's really serious. But so that's what we've been saying, less than 60 is pretty low.
But not zero.
Not zero. But I'm sure, it gets lower and lower as you get younger and younger. I think they stratified that here as well.
They did, and I would like them to change it by 1 year because that would change where I am in the.
Yeah.
So this boggles my mind. 70% of cases asymptomatic.
Yeah, and below 60.
Under 60. Well, I'm looking at the overall average here in this table. Table 1. Okay, gives the total of the total cases. 31% symptomatic.
Right.
My God.
Now, last week, the study we did out of China was 20% asymptomatic. So it varies. Okay. It really varies according to the study and the population and so forth, which I'm not surprised.
Still, it's a lot.
Yeah.
Yeah, absolutely. I mean, all of these people were close contacts of people who were known to be sick. So maybe they got a higher dose.
Yeah.
Or maybe there's something about dose going on here. So that might be a reason for differences between studies. But either way, there are a lot of people who don't have symptoms.
Well, and and then I'm sitting here thinking, in a way, this is good news. Okay. Because we could have this fire just raging through the population. We don't even know it. Okay. And then I'm thinking, well, it would be good to have good serology testing population wide so we can see how much there is. But we just learned that that's not necessarily going to tell the whole story because a lot of these people probably won't even be seropositive.
That's entirely possible. Yeah.
They may have a T cell response, but they may not have antibodies.
Yeah.
Okay.
I'm going to head back to the lab and start working on T cell assays.
Make a rapid T cell assay. You can.
There's like no such thing, right, Brianne?
No, there's no such thing.
You could imagine how you might make one, right?
Maybe, yeah. I can kind of think about how I might make one.
I mean, you start same thing as serology. You start with blood, right?
You start with blood, but again, you're going to have to have cells. You can't just have products like you can with an antibody test.
Yeah. So you have this, instead of throwing away the cells, you keep them.
You need to keep the cells.
Or maybe you don't even have to fractionate them. Maybe you could figure out a test using whole blood, right?
Oh, yeah, you probably could.
How many T cells are there in, say, 0.1 ml of blood?
That's a great question that I do not remember the answer to. Let's see.
Because that way we would know. You could take a finger prick and get enough blood to do this. They have a great figure here. Figure 1, percentage of critical and symptomatic infections in males and females.
Yes.
80, 60 to 79, all the way down. And you can see how the symptomatic versus critical goes. It's very, very illuminating.
These two papers together for me are really an eye-opener. This is really something. I'm going to have to think about this. What a story. What an amazing story this is.
So if I'm doing my math correctly, it's 2 times 10 to the 5th T cells per 0.1 mils.
It's a lot.
I have 2 times 10 to the 9th per liter.
Wow.
So if I do my division correctly.
Per liter, 1,000 mLs, yeah.
Yeah.
It's 10 to the 6 per mil, right?
Per mil, so 10 to the 5 per 0.1.
Yeah. 10 to the 6 per mil, that's like HeLa cells.
That's right.
That's pretty dense.
It's very dense, yeah. Yep.
So how many T cells do I need to do an assay, Brianne? We're really pushing on you.
You really are. I think I was usually doing assays with 1 or 2 million back in the day.
And you said 10 to the 5th and 100 microliters?
Yeah.
So I need a mil of blood to do like a well, right?
Yeah.
Okay.
So you think, Brianne, you could take this and add the peptide and measure gamma interferon in some way? Would that be the best way to do it?
I think that given the data that they have here, it seems like that would give us something useful. That would require you culturing the cells with peptide in order to get that gamma made.
How long does it take? A couple of hours?
When you do the flow cytometry version, you usually have the cells with peptide for 5 hours. I think when you do an Ellie spot, like they were doing in the previous paper, it's overnight.
Is there some way you add an inhibitor to keep the protein in the cell from being secreted, the interferon?
So you do for the flow cytometry assay, you add a drug that actually stops transport from the Golgi.
Right.
So that you stop the protein from being secreted.
So maybe that would be something useful because you don't want to have flow on this rapid assay, but
right.
If somehow you need a color change of some kind, right?
Yeah. And so with the Ellie spot, what they do is they have the cells making cytokine and secreting it onto a membrane.
Right.
And then they basically are doing an ELISA on that membrane to see if there are spots where cytokine was produced.
Right. Okay. Well, we've just given people good ideas. Go to it. So let me summarize this. So 5,400 contacts of SARS-CoV-2 index cases in Italy. Positive were determined by PCR or serology. 74% of all infected individuals, less than 60, did not develop symptoms. 74%. The risk of symptoms increased with age, and over 60 years, 6.6% had critical disease. So this is very important. You know, if you are older and you don't work, you should stay home for a while.
That's what I'm doing.
What are you?
Older. I fit all the criteria.
What are you?
I'm older,
you're 80? Are you 80?
I'm not 80.
You're 70s, right?
I'm going to be 72 on Monday.
Oh, happy birthday.
Yes, happy birthday.
That's 2 years from TWiV 500.
Yeah, and you were 70 on for that episode,
Yep.
right?
Yep.
Yeah, and I'm 67, so we're both in the high risk category. So we shouldn't do too much. I mean, I'm not going anywhere. Although today I went to get my eyes examined, and this is so weird that she's sitting 6 inches from me on that weird apparatus, and we're both wearing masks, and I'm thinking this has got to be the most high risk thing that you can do.
Well, my wife's been to the dentist.
Oh yeah.
Because she was halfway through a crown job when this struck. So she was wearing a temporary crown for overtime.
Wow.
And when we came off a lockdown, she went to her dentist, and unless she had an asymptomatic infection, everything's okay. But
well, here you can go to CVS and get a saliva PCR.
Is that right?
Anytime you want. Yeah, it was developed by Rutgers, and the state bought it. And yeah, that's what Dixon and his wife did. And I think you should do it once a week. That would really be informative, right?
Well, yeah. And certainly, I think one thing people don't understand is you can't just go get a test and you come out negative and you say, oh, okay, I was negative.
That's it.
Whether you're negative or positive, that could change next week.
No, you have to go every week because the test may be wrong. But then a week is a good time where you could get infected.
Yep.
And then if you are, you stay home for 2 weeks. And everybody who was in contact with you, you stay home. And that's the way to take care of this. If we did that universally, it would be fine. But nobody wants to do it, or few people want to do it.
Vincent, do you have a plan in your house for what are you going to do when somebody gets sick?
No, I don't have a plan, but I know you do.
Yeah. Well, there's only 2 of us. It makes it easier. Okay.
Yeah, we got 4 people here. 4 people and 2 dogs. I don't know. I'd stay home, of course. But so Columbia opened up this week, but not full time, but more than before. And we had to get this app. This is just crazy. There's this app we had to get. It's called, what is it called? See You Back to Work. Isn't that clever?
Yeah, that's very clever.
That's
good. So you go in every day, reopen CU. Actually, I would have called it See You Back to Work, but they call it reopen CU. And so you every day you go. It's called COVID-19 attestation. See, symptom attestation. I don't know if you can see. It's too bright.
Wow.
Right?
Yeah, it's too bright. Okay.
You go. Do you have any of the following symptoms? Cough. Blah blah blah. Tested positive. Close to someone who was positive, or none of the above. You push none of the above, submit it, and then it turns green. And then when you go in, you got to show it to the security guard, or they don't let you in. but it's just, you could say no to everything, right?
Yeah, sure.
Right, sure.
And that's fashionable nowadays, is to just say, fabricate your own reality. And that's fine.
So then we have all these rules for, you wear a mask in, then you change it and you put on your lab mask. Oh, man, you wash your hands, do this and that. So I just stay in my office. I don't go anywhere. But I do think it's a little riskier. And I see people on the street, and about half of them are wearing masks and half of them not. And a quarter have the mask below their chin.
Yeah.
What the hell is that for, folks?
Right.
Maybe their chin's cold.
So do you go into your office at all, Brianne? You do, yeah.
I do. I go into my office, and I actually am going into the lab sort of Monday through Friday, getting some lab work done. I'm pretty much the only person who I think is in the building, or at least the only person on the biology floor.
You wear a face mask when you go in?
I do wear a face mask because whenever I think I'm, completely by myself, somebody walks by and wants to have a conversation. So I never know when I'm going to run into somebody.
And when you go in your lab, you take it off probably because there's nobody there, right?
Right. When I go in my lab, I take it off. Yeah. But otherwise, I have the mask on, work with my cells, do my experiments.
Yeah. And Rich, when you go out, you wear a mask, I presume, right?
If I'm going out into a store or something like that, where I'm going to have a close interaction with people, but I don't do that much.
If you walk, though,
like today, our refrigerator broke. So today today I had to go out and get ice. So I went to a convenience store and I wore my mask. If we walk around the neighborhood, I don't. But we're way over. We're outside. We're moving. We're way over 6 feet from anybody.
But you have one with you in case someone is coming by you?
Not when I'm walking around in the neighborhood, no, because it just doesn't happen. Okay. If somebody's approaching us on the same side of the street, we cross the street. Yeah. Or go in the middle. We're leaving lots of space. And everybody around in the neighborhood gets it. Okay. And they're all very respectful of that. And we all actually, it's been great because a lot of people are outside on a regular basis doing this kind of stuff. I know my neighbors better than I ever did before. Okay. And we're also getting, I confess, a little looser with my extended family.
Yeah.
Okay. But, so we're sort of playing that one by ear. When my grandkids come over once or twice a day to visit, we sit outside. Okay. We don't wear masks, but we sit outside and we're fairly distant from each other. Oh yeah. It's cool.
I designed that.
Very nice.
Very nice. I have my own, I made my own masks. Got my homemade mask. Oh, very good.
I like it. Well, I always wear a mask even when I walk around town. I've noticed that my pollen allergies seem to have gone away when I started wearing masks a lot. So cool. I'll wear masks all the time now.
Actually, for a while, when things were letting up and before Texas started to spike again, I went rowing a couple of times. Okay. And that I followed their rules, which is when you're on the dock or getting your boat down or that kind of stuff, you wear a mask. Then when you actually get out on the water and there's nobody around and you're outside, you can take your mask off. And that's, if I were going to go walking downtown, I would wear a mask. But I don't. I haven't been walking downtown in 3 months. And now that the Texas is starting to burn, I don't even go rowing anymore.
Texas is starting to burn.
I can wait. I can wait.
Let's do some email. Who wants to take the first one? I don't.
Okay. Nathan writes, Dear TWiV. Okay. Yeah. I have a young grandson, so I can get into that. My 4-year-old son often plays while TWiV is on in the background. Someone on the show mentioned fecal-oral transmission of SARS-CoV-2 a while back. He asked what that means. My 4-year-old son, asked what that means, and I told him. Since then, he has been begging and badgering me to send a specific question in to you. I've resisted so far, but now I relent. Is there any chance that SARS-CoV-2 can be spread through farts? That is all. Thank you, Nathan. That comes from his 4-year-old son. I get it. Great question. Yeah, why not?
I would think probably not, because you have, some cloth coverings.
That's true. But, 4-year-olds walk around the house and maybe they're naked and farting, right?
Maybe.
But,
I would say in the absolute that if there is, this is all extremely low probability because, we don't even know for sure I guess there have been incidents of, bad plumbing spreading virus.
Yeah.
Okay. Which is evidence of fecal-oral transmission. We've seen infectious virus in feces. So although epidemiologically, I don't think that there's good evidence that this is a major route of transmission, it's a possibility. And so if you've got virus in your gut, it seems to me that if you expel gas from your gut, there's not a zero probability there's going to be virus in that. I don't know.
That makes sense.
Is there such there are respiratory droplets? Are there GI droplets? Sure. Why not? I don't know that anybody's measured it.
Interesting. Certainly when
we
you
need
have
to use the Gesundheit machine backwards, right?
That's an interesting point. I would say it's very low because fecal transmission is pretty rare, even though and it's associated with plumbing and fecal plumes created by toilets. So I think unless we're making fecal plumes, it's an interesting thought, right? I think we're not. I think it's very, very low. So I would say probably not. I think that
you really need a 4-year-old male to ask the question.
It's true.
I know all about
probably
this.
True. Yeah.
Little boys are all about farts.
Yeah. All right. Brianne, you want to take the next one?
Sure. Bill writes, "Hello, everyone. How risky do you think air travel is right now, assuming that social distancing is taken into account and masks are required? Would any of you fly on a plane? Why? Why not? I've read a couple of articles claiming that it's relatively safe because the cabin air is frequently replaced. However, in both cases, this information came from airline industry talking heads, so I'm skeptical. Thank you, Bill."
Okay, I got heavy into this one.
Okay.
So I'll give you my wrap because I see this as I see this as a potential teaching moment in critical thinking, which in my view is part of what TWiV is about. My first reaction to this was a graph that I had seen on Facebook. I confess it was on Facebook. And I put a link into this graph. It's the first one down there. And it's an infographic that shows that purports to show sort of relative risk on a scale of 1 to 10 of various activities. All right. And just to give the bottom line for Bill, it puts airplanes smack dab in the middle. And it equates, it's the same category with a backyard party, going to a beaches, bowling, dinner parties, malls. But then here's the teaching moment in critical thinking. I look at this and I say, okay, graph on Facebook. What does that mean? And so I do my homework. And first of all, I want to congratulate Patrick Broom, who made the infographic, because not only is it a nice looking graphic, but he has the sources here talking about how the how the information was gathered. And he has a nice summary of it. And then he's got his sources up the top. First of all, he links his own site. He's a graphic artist. Okay. So he knows not necessarily anything about the science, but he turns it into graphical information one sort or another. And I looked at his site to get an idea of that. And that's where I got the original figure. And then he gives a link to an MLive article that the information came from. So if you look at that, there's an article where MLive, which I looked into that as well. Okay, what's that source? Okay, and it's a news outlet that's online, but is responsible for a number of newspapers in Michigan and elsewhere. I looked that up on Wikipedia. Reasonably reliable. And the deal here is that this fellow interviewed 4 different experts, if you like, a Beaumont Health Director of Infectious Disease Research, all of them with doctor in front of their name, McLaren Health Center Medical Director for Infection Prevention, retired infectious disease specialist from Spectrum Health, Henry Ford Macomb Medical Director for Infection Prevention. So these people know a little bit about infection and disease and contagion. And they gave them 36 different activities and asked them to rate these on a scale of 1 to 10. The doctors used 5 different factors in assisting their judging, whether it's inside or outside, how close you are to people, exposure time, likelihood of compliance, personal risk level. And then there's a long wrap on each one, summarizing the risk and with a few comments. So this is, I would say, informed opinion.
Yep.
It's not data.
Because we don't have any experiments to check this, right?
No data. No data. And the graphic has, it's appealing because it has an element of truthiness to it. If you look at these different categories, they seem to make sense. And in fact,
kind of.
Okay, so you got a gripe about some of these, Brianne?
There are a couple places where I'm a little uncertain. And the one that really stands out to me is the dentist. Which it puts as somewhat low risk compared to some of the other ones. And I remember our previous conversation with Michael Schmidt and things like that about how all of the tools are creating aerosols and they're in the mouth and you're in close proximity to one another. And so I wonder about dentist being quite as low as it is.
Right. That's category 4, one below airplanes. Okay. So, those are that's the best I can give as a way to sort of address this. That's the sort of information I have. I find this useful, but I approach it with caution but I know what the sources are because I looked into it. Okay. And to give you my bottom line, I'm not flying anywhere.
Yeah, I wanted to ask you that. Okay. What about you, Brianne?
I'm probably not flying anywhere. I haven't had to think about it, but I have had to think about trains. And I have chosen to not ride trains. And I think trains actually have less air circulation than a plane. So I've definitely chosen to not ride a train recently. And I am not planning to fly anytime soon.
How about you, Vincent?
Yeah, I'm not flying. Everything I was going to fly to is canceled for the rest of the year. So that's it's easy for me to say if I had to go, I don't know. But I think I just don't trust planes. I don't trust that everybody's going to wear a face mask, right?
Right.
Or they're not going to take it off to eat their peanuts or something.
Plus, it means you're going to be in an airport. And, on a plane, respiratory viruses typically spread around the person who's infected, not throughout the plane. So there's less issue with air circulation, but it's just around you. So who knows if you've got someone infected near you. But really the key is that I'm in the high risk for serious symptoms, right? If you're 20, I think you should factor that into your decision. You're not likely to have serious symptoms. And if you need to fly somewhere, you could do it. But if you're 90, I've gotten questions. My 90-year-old grandparents want to go visit their 2-month-old grandchild in California. I would not do that. I realize 2 months happens just once, but you only live once also. So 90 is way too high risk to be doing anything, in my opinion. Anything 70 or above, it seems to me you better stay home as much as you can.
I answer that question with a similar sort of caveat of I haven't really had the question of having to fly anywhere. I don't have any place I would fly isn't going to happen. I suppose if I absolutely had to fly somewhere, I could make sure I was wearing my mask and see if I could be seated as, far from other people and not sit near people in the airport and wash my hands a lot while I'm in the airport and use hand sanitizer frequently while I'm on the plane. I suppose I could do all of those things if I had to fly, but I don't have any reason to have to fly right now.
Yeah.
Yeah, I pretty much agree. And the other thing that I would add is with respect to myself, I don't want to get sick on the road.
Yeah.
Okay.
Yeah.
I figure there's a good chance I'm going to get sick. I figure there's we can talk. Actually, we'll have an opportunity later on if we get to the emails to talk about herd immunity again. But I figure there's a pretty good chance that I'm going to get sick, and I want to get sick at home. All right. I don't want to be on the road. It would just be inconvenient. It's going to be a lot easier to do if I'm home.
We'll have more on this. A lot of questions. One of them coming up is about playing basketball. I noticed that was on. That was on one of those.
It is, and it's relatively high risk.
Yeah.
It's group 7 out of 9 in terms of high risk.
Okay.
Right up there with schools and public pools. That's the big one to me. August, schools are going to open.
Yeah, but they're kids, They get, like we saw last week, they get infected less. And
fine, but they're going to cook it up and bring it home.
They could cook it up and bring it home, yeah.
Yeah, I don't know if they're going to comply with the rules.
No. Actually, in Texas, I don't think they're going to require masks in the schools.
Oh, that's crazy. That's crazy. If you're going to go to school, and some places are going to let people go to school, you got to wear face masks. If you don't, you're playing with Russian roulette or whatever. Okay, Carolina writes, greetings from Eastern Connecticut. Currently 33C, 92F, 167% humidity.
Yep.
In relation to the C versus F debate, I feel compelled to weigh in that the clear advantage promoting the comprehensibility of the Celsius scale is that it is freaking linear. 1 degree is worth the same regardless of where it falls, which is clearly not the case for Fahrenheit. Also, from a scientific point of view, Celsius and Kelvins are highly compatible as each step on the scale is worth the same. And for conversion from C to K, you simply add 273.15 rather than multiplying with 9/5 and adding 32 for F. I'm a Scandinavian explant in the U.S., and the widespread use of it, the Fahrenheit scale, is likely the most prominent obstacle to my integration into this country, perhaps along with the U.S. customs and immigration system. However, that's not my main reason for writing. I could not avoid reacting to the question in TWiV 628 regarding potential viability of SARS-CoV-2 beyond death of the infected individual. That's what I love about TWiV. We say we don't know, who knows, and someone answers.
Somebody writes in.
Yeah, I'm expecting someone to really correct all of my numbers about those T cells from earlier.
Yeah, we ought to get a lot of letters about farts, too.
Maybe John Udell. I work with foot and mouth disease virus, clearly a very different virus. Perhaps specifically it is non-enveloped. However, FMDV is not at all durable outside the infected host, specifically as the capsid is highly unstable outside a pH of 7 to 7.5 and therefore quickly disintegrates in most environments. Due to food safety concerns, several studies have confirmed that FMDV quickly becomes inactivated in muscle tissue, i.e. meat, of infected animals due to post-mortem acidification, rigor mortis. However, the highest viral loads in infected animals are found in vesicular lesions in peripheral epithelial tissues, the feet and the mouth, hence the name. Due to the apparent lack of information regarding FMDV viability at these peripheral lesion sites, we recently conducted a largely opportunistic study to investigate this.
Excellent.
We initially utilized carcasses from pigs that had been part of an experimental study during which they were humanely euthanized at the peak of clinical FMD. Carcasses were stored at 4 degrees C. FMDV is a select agent, and our options for prolonged carcass storage were therefore limited to a designated cooler room. To cut a long story short, we were very surprised by our findings and had to repeat the setup with another 3 batches of unrelated FMDV-infected pig carcasses before we had to call it quits. Although we did confirm that the virus was quickly inactivated in muscle, it was clearly perfectly happy and well for at least 11 weeks post-mortem in the vesicular epithelium, while still attached to decaying carcasses. I can confirm that the pigs were definitely dead, as I had to visit them once weekly for sampling through 11 weeks sparing only Christmas Eve. I apologize if these details are maybe a bit morbid for the average listener, but knowledge concerning potential viability of this highly infectious agent in decaying carcasses is critical in relation to practical handling of deceased animals during outbreaks of FMD. So I guess the take-home message might be that some RNA viruses can apparently remain viable through prolonged periods despite the death of their host, as long as the environmental conditions are favorable. In this case, this implied relatively cold and humid storage and distant from the putrefaction that was inevitably happening within various body cavities. Thanks for all you do. Carolina is a DVM, PhD, and a picornavirologist. Yay! She sends a link to the article which she talked about. Okay.
That's outstanding.
That's really interesting.
I think that I'm sure someone is doing this for SARS-CoV-2. In fact, I know some people I could write to and ask if they're doing
certainly hope so. And I think that this highlights the fact that they need to think about different parts of the body. It's not just one anatomic location, because she says that the virus went away quite quickly in the muscle. But when she looked at other tissues, she was able to find that virus.
So there's going to be a lot of variables here. The type of virus, where you sample, et cetera.
Yep.
So she's I'm looking at this paper, she's at Plum Island.
Wow.
Is that still operative?
It is. It is still operative.
Because that whole operations, she lists also Kansas State University in Manhattan, Kansas. That's where Plum Island is going to move, right?
That's right.
They're building that big facility there, but I guess that's not, do you know if that's operational yet?
I don't believe it is.
Maybe she can write back to us and tell us the status of that whole thing. Have you visited Plum Island, Vincent?
I have. Yeah,
I think we've had this conversation. Yeah,
a couple of times. It's interesting. And in fact, someone asked a couple of years ago to go out there and do a TWiV. Just never got around to it. Should do it before they close.
That would be really cool.
I know some people out there. Brianne, you could come.
I can come. I just won't take a train.
We could drive out, right?
Yeah, I would drive.
Although you and I can't be within 6 feet of each other.
No.
Oh, man. You can sit in the back.
We can both drive and sit far apart.
Oh, you could drive your own car. Is that what
I can drive my own car.
Okay. Well, then in that case, you'd get there faster, because I understand you're a fast driver.
You really?
Well, you told me. Yeah.
Oh, okay.
Rich, can you take the next one?
Barbara writes, I guess this is says 623.
Yeah, I started to date the emails because
okay.
You know.
Dear Vincent and TWiV team, I'm a writer specializing in communicating environmental and wildlife conservation in Canada, Mexico, and the U.S. I've been listening to your show since mid-January, well before we thought SARS-CoV-2 was going to turn into a pandemic that would forever transform the world as we knew it. When I first started listening, I thought it was Alan Alda's podcast. He has a medical podcast, apparently. And because Vincent sounds more like Alan Alda than Alan Alda, I was left gobsmacked by how much the American television and film actor knew about viruses. Very good. It wasn't until the end of the first show that I realized Vincent was not Alan Alda, but it was too late. I was hooked. To me, you are Alan Alda, Vincent. I am writing with a question for each of the TWiV hosts and also Daniel Griffin, if possible. Based on what you have learned about SARS-CoV-2 over the last several months, are you more or less afraid of catching the virus? And we'll come back to that. In closing, I self-identify as a hypochondriac. Is that a gender? A hypochondriac who and have been acutely aware of SARS-CoV-2 since mid-January, when the WHO was stressing that countries needed to pay attention. Yes, the WHO was telling this to the world way back then. Canada, my country, did not step up to the plate until the second week of March, long overdue, but formidable coordination and cooperation by the national government and the provinces nonetheless. As a hypochondriac, my advice to the pandemic planning teams around the world for the next time is that you may want to consider having one of us, a hypochondriac, on your response committee. We would have had people mobilized for physical distancing, wearing masks, and hand sanitizing months prior, and we would have saved a lot of lives. if you're ever in Guelph, Ontario, Canada, my hometown, drop by for supper. The yard is big, and there's always room for one more. University of Guelph is famed for its veterinary school. In fact, there's quite a bit of current SARS-CoV-2 research underway, and she gives a link to a Guelph news site and thanks us. Yeah, I have known scientists, Canadian scientists from Guelph. I think Dave Evans started off in Guelph before he went to Edmonton. And if I'm not mistaken, Guelph is the home of the RoboGut. Okay. Yeah. Where the RoboGut is, about the technology of fecal transplants?
Yeah.
Yes.
And the RoboGut, fecal transplants are usually done with material from fecal material from a cohabiting individual, somebody who's likely to have a microbiome similar to yours. But the RoboGut is a method for trying to generate material that is like fecal material in terms of its microbiological content and stuff. Okay, back to the question. Based on what you have learned about SARS-CoV-2 over the past several months, are you more or less afraid of of catching the virus? I will answer for myself. Based on my understanding of how this works, I have been resigned to catching the virus eventually from the get-go. I figure that we're going to reach, barring a vaccine, and it's a race between the virus and a vaccine, and I think it's likely to be a tie at best. I figure that we're going to reach herd immunity at about 80% infection, which means that my chances of getting infected are about 80%. And I've been resigned to that from the get-go. It's not a matter of if, it's just a matter of when. I regard myself, even though I'm in a high-risk group of 72, as actually quite healthy for my age. So I don't, of course, there's a lot of denial here and a lot of Pollyanna stuff. But I figure I will get probably sicker than I've been in a long, long time and won't be hospitalized. And that'll be the end of it. And sometimes when I get really close to that, I get a little bit of a twinge, like, ooh. But mostly, and it hasn't changed from the get-go. Been the same for me the whole time.
So you're not afraid, right?
I'm not afraid.
Yeah, but you're a tough guy.
Well, I'm better with things that are actually happening than I am with imagining things. And I think that's probably one of the reasons that I'm comfortable with this, because I figure it's effectively happening. I may as well be sick, okay, because it's going to happen.
Yeah, it's interesting.
Now,
I didn't think of it that
talk
way.
to me again when I start to get sick. Then I may get a little scared.
Okay, you can come on TWiV when you're sick, okay?
Yeah, okay.
Brianne, what about you?
So I think that I was originally sort of feeling like I was worried in that I didn't particularly enjoy having H1N1 and didn't want to go through something in any way similar to that again, but also realized that due to my age group, I probably wasn't at a huge risk of severe disease. So I wasn't, excited about it, but maybe not terribly concerned. The more I hear about things like some pathological changes to the lungs in people who may have had asymptomatic infection or things like that, the more I'm realizing that this virus may be doing some things that are not so great, and I would prefer to not have it if that is at all possible. And I think before I was a little more lax about that, but now I would really like to avoid it as much as possible for myself. Before I was avoiding it for spreading to others.
You're up, Vincent.
I'm not afraid. No, I'd rather not. But Ian Lipkin is my age, and he got through it. He said it was a rugged infection, and at one point he kind of was getting short of breath, but never had to be hospitalized. And I think I've generally done well with infectious diseases, so I have a sense that my genetic makeup of my immune system is pretty good. So I think I would do all right. So I'm not afraid. I could die, yeah, sure, and that would be sad because I'd miss a lot of things like TWiV, and it would probably end if I died, unfortunately, because nobody else would want to carry on. But I don't think that's going to happen. Although Yeah, a virus taking my life, boy, that would be something, right?
Yeah.
That would be ironic.
It's like a life of viruses and then a life ended by viruses. That would be, someone would have to write that, I guess. So, no, I'm not afraid.
I'll write it for you.
But I am careful. I try and be as careful as I can. I mean, I do go to Columbia twice a week, but I drive and I really do not encounter anyone. I do minimal, the eye doctor was the first time I'd been in a small office in a long time. The only other place I'd been to was a liquor store, really. That's the only thing I would risk buying some wine. Go buy some wine. But there's nobody in them, and you can wear face masks. So I'm careful to wash my hands and so forth. So I don't want to be infected, but I'm not afraid. I don't know what the other TWiVers would feel, Be interesting to find out.
Yeah, I think you pretty much described my situation. And in terms of taking precautions, it has as much, if not more, to do with other people than it does.
Yeah, I think
with
so
me.
too. Yeah.
I think to me, the critical factor is people, the whole flatten the curve meme has sort of dropped out. We don't talk about that anymore, or not much anyway. But to me, that's still relevant. The whole deal here is to try and mitigate in a fashion so that the healthcare system doesn't get overwhelmed. Okay. Yeah. And we all have a responsibility to participate in that. That's a group activity, and it's everybody. Everybody has a responsibility to do what they can to mitigate the spread of disease. Yeah, you're probably going to get it eventually unless we come out with a vaccine, but we don't want to all do it at once, or it's going to be a lot worse for everybody.
Absolutely. And you never know, who might be immunocompromised or who might have some type of thing that could make things worse. I don't know if it's because I'm an immunologist, but people seem to like to share their stories of being immunocompromised with me. And so there are a fair number of people who I know who have something going on with their immune system. And so I think about them a lot when I'm thinking about whether or not to take precautions. That has allowed me to have some very salient images in my mind of who exactly I'm protecting as I'm wearing my mask or taking my precautions.
Yeah, I think that's important because, young people, they want to go out, they want to go swimming, they want to go to bars and do stuff, and they do it, and they're not going to get very sick. The numbers say they're not going to get very sick, right? But who knows when you're going to encounter an older person who will then get very sick. You just don't know that. And that's what you should be careful about. I want to go resume my life. I was talking to a journalist the other day, and she lives in New York, and she said outside her building, which is in Lincoln Center, she said there were these people from New Jersey. I said, how'd you know they're from New Jersey? She said, the woman's hair was the color you can only get from a bottle. Boy, if that isn't stereotyping. Anyway,
yeah.
They weren't wearing masks. So she said, wear a face mask. And she said, the woman said to her, it's a free country. I can do what I want. So that's not right. You don't have the right to make other people sick. That's not the right attitude, folks. Anyway, Brienne, can you take the next one?
Sure. Jerry writes, maximum respect to Vincent Rich et al. This retired physicist, who studied microbiology and biochem too, is addicted to TWiV. Thank you. It's the great weather season here in Seattle, mid 75 degrees Fahrenheit and dry. Here's a photo I took yesterday from a block north of my apartment.
Man, that's gorgeous.
That's a gorgeous photo.
You got the Space Needle and Mount Rainier. It's that is a gorgeous day in Seattle. It's not often you can see Mount Rainier that clearly.
That is beautiful. As you know, there's now a massive outbreak in Germany, my other home, at the Tönnies factory and community in Gütersloh. Where's Kathy to pronounce the German when I need her? Of course, the people work close to each other and engage in vigorous physical activity. But there are other jobs under such conditions. There must be an environmental component, cold, damp, fog in the refrigerated environment, providing a long-lasting suspension of virus-bearing droplets. Thanks again for your thoughtfulness, even the prissy grammar and punctilious comments about what can mutate and what cannot. Excellent. I would agree that we should think more about the environmental components that are important for virus spread. I note that he says something about the damp fog in the refrigerated environment. And I know that from some of the data I've seen, humidity seems to be particularly important. I am always struck by one paper from Akiko Iwasaki's lab about humidity and virus transmission that I read with my students, and I go back to every time I think about environmental conditions.
So he's referring to a story about a German slaughterhouse.
Yeah. They've had an outbreak
that an article I got here of over 1,000 cases. This slaughterhouses seem to be a place where this happens. And yeah, it does leave you wondering whether there are environmental conditions that contribute to this, but there's so many variables. I've never been in a slaughterhouse. Have you?
No.
I have not.
So we've had letters from people who have we not?
I don't know.
So I'd be interested in hearing from somebody who has a lot of experience with a slaughterhouse, who could tell us kind of what goes on and what the variables are there that might contribute to spread. Because I imagine it to be a place where there's people in fairly crowded conditions and lots of opportunities for aerosols, but I don't know.
Right. He mentions the cold and damp here, and I would guess that there was probably not a lot of air exchange with the outside.
Well, damp is going to make the droplets bigger and fall to the ground, so I'm not sure that's facilitating it. The low temperature may, but I think it's mainly lots of people working close and not being very clean, right? And opportunities for contamination, because it's happened many times in the U.S., right? That's one of the major outbreak sites in the U.S.
Yes.
So I don't know. I mean, how many other work environments such as that one are actually active right now, right? This is meat, so it's food, it's essential, and so it's going on. But are people still building cars, for example, right? Which is, I guess, a lot of people on an assembly line. I don't know. What other things, any kind of food production where there are a lot of people involved? We don't hear about outbreaks.
We don't.
I just don't know. The problem is there's no data, Dr. Glomf. There's just no data. All right, but it's a good question. Doug writes, hi there. If an asymptomatic person is in a small gym on a treadmill and presumably breathing hard, she will be shedding via droplets and aerosols into the air, correct? Yeah. These droplets can stay in the air for X number of minutes, hours, correct? Air circulation in the gym would move these droplets around, increasing the area affected. All this is true. Those who could not find an N95 mask but instead wear a homemade or mass-produced economy mask will be at considerable risk, correct? Assuming these lesser masks are lucky to be 60% effective. The same logic, but to a lesser degree, would be true in a restaurant, right? Your insight would be greatly appreciated. All right. I'm not sure where you get these 60% effective. But a good cloth mask with a thick cloth that can pull out these droplets will be quite effective. And you don't have to be 100%, right? Because it's all about dose with viruses. And so if you don't breathe in enough droplets to infect you, you won't be infected. So I think even an economy mask, as long as it's not very thin, will not put you at considerable risk. However, the risk here is being indoors. I mean, we know most of the infections happen indoors, where people are congregating, right? And so if you're on this treadmill, there are presumably other people around. That's a high-risk environment. If there are other people around. If there's no one around, it's obviously not high risk. So I'm not so much worried about the mask worn properly as the environment.
Yeah, I think it's also hard to tell here if the person on the treadmill is wearing a mask or not. I guess it is possible to wear a mask on a treadmill.
Yeah.
He makes a comparison to restaurants, and I think you are less likely to wear a mask in a restaurant, as that would sort of make eating hard.
Yeah, at some point you have to take your mask off, and that's why they space the tables out, right? And that helps, but as we saw in a paper we did weeks ago, the air vent above a table can recirculate virus and bring it back down onto you. So,
So in my earlier truthy infographic, gyms are up there at 8, along with amusement parks, buffets, churches, sports stadiums. And the comment from the panel of experts are, similar to singing, people emit more respiratory secretions when working out and breathing heavily. It's also tough to work out while wearing a mask, can be difficult to distance. Once gyms are open, one of the experts suggests using individual equipment and wiping it down instead of doing indoor group activities like spin and dance classes. Our gym, I haven't been back, and that was a regular activity for us. You now can only, because it has they've limited the capacity to limit crowding, so you have to sign up for classes. We used to do yoga and body pump, and I used to swim. You can't use the locker room. If you want to swim, you can sign up for a lane, and you go in your swimsuit and leave in your swimsuit. You can't use the locker room. You can use the toilet, but not the locker room. I would imagine they have a grid on the floor to identify personal space. You have to wipe down your own equipment. You have to reserve space so it doesn't get overcrowded. It's just too much of a hassle. I'd rather go for a walk.
I think you have to, if your places are open, you have to be careful. That's why you're asking this question. I understand. I would not go to a gym on a treadmill. I think the risk is too high. But if you're 20 and the gym is open, I think it's less risk for you because you're not, again, it's all, you have to add everything up.
But if you get sick, you're going to spread it around to somebody else, maybe.
Yeah, it's true. It's another consideration. Rich, you're next.
Barbie writes, love the show and listened in the car while driving to northern Minnesota for a very socially distant trip to Voyageurs National Park, where the weather was nice, but we had 1 leech, 9 ticks, and 1 billion mosquitoes. You discussed 2 Nature medicine papers in this episode. The epidemiology study suggesting kids are infected less often, and the study showing asymptomatic carriers may not generate an antibody response. I was waiting for you to loop back and discuss this in the context of what might be happening in the kids. My question, if kids are infected less often and it's because of some form of cross-protection from another coronavirus, would you expect them to retain any form of immunity as they get older? Also, might this affect their ability to respond to vaccines? This is concerning because we likely won't know the answer to this until much later, since the trials won't likely include children. Thank you, Barbie. I don't remember, I only vaguely remember the notion that kids are actually infected less often. Did we discuss
that was that was Friday. You weren't there, but our paper on Friday was indicating that it was a statistical analysis, right? It was modeling based on the available data, but it suggested that kids are infected less often, yes. But it's not clear that it's from cross-protection at all from another coronavirus. I don't know what the reason is for that. And I wouldn't so I don't think it's cross-protection. I'm not aware of any cross-protection between seasonal coronaviruses and SARS-CoV-2, at least at the level of neutralizing antibody, right?
Right. So this is really hard because we would need to look at T-cell responses, but we would also need to have a population of people who have never been exposed to seasonal coronaviruses.
Which is impossible. They don't exist, right?
Right. And so it's hard to find that cross-reactivity versus not.
The population that is least likely to have been exposed to seasonal coronaviruses is the children, which is contradictory
That's absolutely right.
The study we're talking about.
Yep. Yeah, that's exactly right.
I find that I have perhaps an unreasonable bias against this whole idea that children are not getting infected as frequently anyway. I want to see confirmation of that. Experimental, not models. I did listen to that episode. There was a rap about it.
I was thinking of that, and The problem is that the school's closed early on, so we can't do those studies, right?
Right.
But that would be very interesting to do. And she asks, might this affect their ability to respond to vaccines?
I think if anything, if we're imagining this cross protection, it might be helpful. We might be able to boost that protection with vaccines. I think she's asking whether the immunity would sort of destroy vaccine elicited immunity, but I don't think so. I think this would actually might help out. We might be able to boost that immunity with a vaccine. But of course, we need to look at those T cell responses a lot more before we could address any of these things.
Do you recall in this modeling study what the definition of a kid was, age-wise? Because the other thing that I think about is maternal immunity, passive immunity to the kid.
Sure.
But that only lasts for maybe 6 months or so, right?
Yeah, that's going to be, within the first few months.
No, it wasn't that.
There are older kids than that, right?
So it's age-dependent effects on transmission and control. And so they're looking at 10 to 19-year-olds.
Okay. By then, they probably had the
most of the seasonals,
most of the regular coronaviruses as well. So
the problem is that you get your first seasonal corona infection as a kid, and then you get infected over and over into the rest of your life, right?
Mm-hmm.
So it doesn't make any sense why the kids would get infected less frequently based on immunity of any kind, right?
No, not really.
No. Brianne.
Sure. Marcos writes, Dear TWiV, I write from Spain, where we've recently opened our borders with the rest of Europe, or most of anyway, and will open to third countries soon. Right now, travelers are checked for fever and asked if they've been sick with COVID and visually examined. I don't know the extent of this visual exam. The government said that no PCR will be done to these visitors unless fever or other symptoms are detected, because that kind of massive testing at arrival won't be effective to control imported cases. Even if you don't detect every infected person at entry point because of the limitations of this kind of testing, sensitivity, detection of RNA instead of active virus, etc., wouldn't it be useful? Both my scientific and English background lie very far away in time. Sorry for any blatant mistake I made in these short paragraphs, and thank you for your great work. I started listening recently and will continue to do so in the future. That's from Marcos in Madrid, Spain. I agree, Marcos. I think that the PCR would certainly be useful. And we've talked about data here about percentages of people who are asymptomatic. The paper today talked about 70% asymptomatic. And so you would not catch that 70% of infected individuals.
I wish we had a widespread, almost instantaneous nucleic acid detection assay, right? Do you remember we did a paper a long time ago about CRISPR-based detection, which you could do in the field in a single tube?
I don't recall that, but yeah, well, okay.
One of them was called Hercules. There are a couple.
Oh, right. Yes.
I mean, I'm sure people are working on that now, but I bet you could do a pinprick or a nasal wash or saliva. You throw it in, look for a color change in a minute for nucleic acid. I think that
or an antigen test. Why not?
Yeah, absolutely.
Any sort of quick test.
So. I guess if the RNA's in saliva, maybe some antigen would be too, right? Yeah.
I'm guessing. I mean, this is strictly a guess that the government doesn't want to PCR test everybody just because. I mean, it's a lot of testing and a huge hassle.
But if it's instantaneous, but if it's instantaneous.
If it's instantaneous, if you could do that.
We don't have it yet, but I think we should work on it.
If you could,
that would be perfect.
That would be
or
an
antigen test. Yeah, I agree.
Somebody comes in, if you can tell in 5 minutes whether they're positive or negative, fine.
Because, the paper we just did, 70% of the people don't have fever.
Yeah.
Yeah.
Yeah. So frankly, it's almost as if, the screening that they're talking about is going to catch a minority of the cases coming in.
Totally.
I guess the problem is if we imagine that you have to wait around for, what do we say, an hour for a PCR, a couple hours to get that test done. That's, everybody sitting in a big room for a couple hours. waiting to find out whether they are infected or not, you might see some spread with all those people jammed together.
Yeah, but you could figure out other ways.
Yeah, you have a whole quarantine area, much like, it's immigration is what it is. Okay. And, well, geez, these people, they get swabbed, they go stand in a bunch of lines and doing all their other stuff. And then at the end, they get pulled out. But in the meantime, they've spread it to everybody else in line. Tough. Yeah. You gotta hold them there for that hour.
That's why the fever gun is beautiful, because bung, 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 right?
Yeah.
Mm-hmm.
Okay, you're good. No, off to the side. Swab in your mouth. Can we look at the color in a minute? Less than a minute?
That would do it.
That would be nice.
All right. Mona writes, FYI, the data on next strain includes 113 SARS-CoV-2 RNAs. Sequences about 2/3 are G and 1/3 are D at codon 614. 416. Amino acid 416 of spike.
No, 614.
Is it 614?
Well, that's what it says here.
Yeah, I know, but I don't think that. Well, that's what the next strain is saying. Yeah, 614. Okay, fine. 614.
This is that G614D mutation we've been talking about, right?
Yeah, that's right.
Mm-hmm.
So so most of the sequences, at least out of these 113, are it. Yeah. What that means, I don't know. Could have been just something that popped up early and it's there, right? We don't have any data one way or the other, despite what you're finding out in cells and culture in the laboratory. Rich, you're next.
Darren writes, Dear TWiVers, I'm a computer programmer, but I've been interested in microbiology for some time. I've listened to one of Vincent's virology classes, and I've been listening to and enjoying TWiV, TWIM, and Immune for years. In TWiV 629, Alan expressed a concern that political forces in the U.S. might cause a SARS-CoV-2 vaccine come to the market that has less than ideal effectiveness. If a person gets a dose of such a vaccine and a more effective vaccine comes out, would it be a good idea for that person also to get the second vaccine? Would it be safe? Do vaccines based on different platforms have a booster effect on each other? Thank you so much for your tireless dedication to the science education. Darren, good questions. My knee-jerk reaction to that is that if you get a crappy vaccine and a better one comes out, you get the better one.
Absolutely.
Okay? And you get a boost. I don't see a downside, not obvious based on no other information.
If the second vaccine contains the same epitopes as the first vaccine, then yes, you should get a boost.
So assuming the backbone is different, that would be okay, right?
That would be okay, yeah.
Because if it's like an Ad5 spike and then it's crappy for whatever reason, and then a better Ad5 spike comes out, that might not be good.
Yeah, that could be a little complicated if they were both Ad5.
But I think there's no risk. There's no danger. He says, would it be safe? I think it's fine, right? So I don't see any downside.
I can't think of any downside.
There are no a priori downsides that I could imagine.
Well, and I think of a practical example. I got Zostavax to vaccinate myself against shingles many years ago. Several years ago, and then they came out with Shingrix, which is a better vaccine. I got that.
How you feeling?
Okay. So far, so good. Don't have shingles.
That's a good example. Yeah. Okay. Brienne.
And as a matter of fact, as I recall, we all initially got IPV.
Yeah. Then we got OPV.
And then when OPV came out, they gave us all OPV.
That's right. I did too. I got both. Yep. There you go. History. There's nothing like history, is there? Brienne.
Important to know.
Brienne.
David writes, Hi, TWiV. While listening to TWiV 621's discussions of the different vaccines in the pipeline, I was wondering whether you could see a situation where people are told to get 2 or 3 or more different COVID vaccines as part of their regular prophylactic care, especially if their modalities differ. I don't mean to get them years apart, like how I got the old shingles vaccine a few years ago, and then my doc recommended getting the new one recently. I mean where people get vaccine A this week and vaccine B a week later, or even in the same injection. is anything like this done with another vaccine? Thanks in advance, David. And David is from Teaneck, where the temperature is too hot.
So one of the things that comes to mind is that several of the vaccine protocols for HIV are prime boost protocols that use different platforms.
Yep.
Where they'll prime with, a DNA vaccine and boost with a viral vectored vaccine. So this is not an unheard of situation. And it could be that in order to get a good immune response, ultimately something like that may be proposed.
Sure.
Yeah, absolutely. He mentions getting one this week and one a week later. And I think that's something immunologists are trying to figure out a little bit about, is what that spacing might be, when you might get memory cells, and when might be really good for that second one to happen.
Yeah, because you want to boost a memory response, right?
Exactly. So I don't know of any situations where there are multiple modalities that are done together in the same injection, though there are, of course, times where maybe a couple of different antigens are put together.
Okay.
What would be the memory response? We're talking 2 weeks? Actually, my Shingrix was a prime boost with the same thing, right?
Right, yes.
And that was, they wanted me to come back in like 2 to 6 months, something like that for the boost.
Yeah, I was going to say, when we do the experiments, it's usually a couple months.
Okay.
Judge Brianne, when you get an immunization, you get some memory, right? And then you get a boost. You get a response, and then do you generate more memory cells from that boost?
You do. So the final number of memory cells you have will be more than you had
the first time
after the first time.
Okay.
And those memory cells will be even stronger, better memory cells.
Stronger, better memory. Good.
There we go.
Ron writes, love TWiV. Have been following for several months now, although I should have caught on much earlier. I'm surprised that Rich did not pick up on the understanding that in much of East Africa, Zimbabwe included, many of the adult reproductive age population have succumbed to AIDS, lowering the average age of the population. I think we all did not pick that up.
Yeah.
Because the average age in Zimbabwe is 15.
Yep.
Right.
Wow.
Yeah, AIDS will do it. Have you reviewed the work of Frank Plummer and Alan Ronald, two of my mentors in Winnipeg, University of Manitoba, from 4 decades ago on HIV in Nairobi? No, I can't say that I have. I'm sorry.
I may have, but I can't tie it to those names.
Sadly, Frank, the previous director of the Manitoba Viral Labs, recently died. I believe while back in Kenya, they were instrumental in describing HIV in Nairobi, where they had gone to research Haemophilus ducreyi, and effective at bringing the viral labs to Winnipeg, where your much-discussed Ebola vaccine was developed. I am a simple family physician anesthetist in Sechelt, a small peninsula up the coast from Vancouver, where we have had few COVID-19 cases thanks to the work of our public health department in BC. Our provincial government have been very supportive of the science behind the management of epidemics, although some provinces are a little late to the game. Several TWiV-backed coronavirus researcher noted that a phosphodiesterase inhibitor coded by this coronavirus inactivates interferon to some degree. That was Susan Weiss. This will, of course, be structurally quite different from the multiple found in humans. However, there are a number of medically useful phosphodiesterase inhibitors, including caffeine and theophylline. The most lucrative of these, developed by David Brown in the UK, sildenafil, marketed as Viagra PDE5 inhibitor. Yes, sir. Has he been involved at all in that? He must have some significant background in PDE inhibitor research, which might be of some value in SARS-CoV-2. I don't know. I don't know if anyone's looking at those.
Boy, the idea that Viagra might protect you against SARS-CoV-2 is really scary.
You shouldn't have thrown that out there, Rich. Now,
sorry.
Now there's going to be.
Sorry. Yeah, I'm sorry. I take it back. Edit that out.
That's all right. Too much work. Well, here is a thing I've just typed online, and here's a paper in Clinical Immunology. Any possible role of PDE type 5 inhibitors in treatment of COVID-19? So it's a question. So it's been thought about. Keep up the great work. This has brought me back to my microbiology roots from 4 decades back. Boy, things have changed. Looking forward to more. Ron is from, as he said, Seychelles, a constitutional monarchy, but also a democracy. Is that Canada?
Yeah.
Yes, that's Canada.
So that they follow the Queen of England, right?
You bet.
Yes, they do.
It's a Commonwealth nation. Is that right? Do I have that right?
It is.
These political structures are confusing to me.
Rich, you're next. Do a couple more.
David writes, I'm a high school science teacher from Knoxville. All right. Knoxville, Tennessee, that has been following your show for 2 years. Thank you for making the time to communicate science to the public. You're welcome. We love it. I'm wondering if you could clarify something for me. I understand that an antibody test / ELISA test is used to determine if you have had an immune response to the virus. What is not clear is how we are able to identify if an individual's antibody response is specifically due to SARS-CoV-2 and not some other virus. I find this confusing because I am under the impression that scientists are still not certain which antibody inhibits the SARS spike protein response for entering cells. In addition to this, from what I have read, scientists are experimenting with multiple monoclonal antibody treatments that could act in unison on the virus. Do the monoclonal antibody being explored mimic the amplification of antibodies seen in our serological assays? More simply, are we able to assess whether an individual has had SARS-CoV-2 based on the distinct types of antibody proteins that we see amplified after an infection? Thanks for your time. Your number one fan in South Knoxville, David. Brianne, you want to do that?
Sure. David, those are great questions. I'm a big fan of your questions. So the first thing that you ask is how it is that with an ELISA, we can tell that the antibody response is specifically due to SARS-CoV-2. When we do that ELISA or that antibody test, we are using a piece of the SARS-CoV-2 virus, a protein like maybe the spike protein, and we're using it almost as bait for antibodies. We're trying to see if a person has antibodies that bind that SARS-CoV-2 protein. And then we wash away all of the ones that don't bind that SARS-CoV-2 protein. And we're able to kind of measure how much or how many of those antibodies that bind the SARS-CoV-2 protein we have. So we know that the response is specific because we're trying to get the antibodies to bind to a SARS-CoV-2 protein. And we can say that we are only going to have kind of a measurable amount of those antibodies if we have had infection. We would have too few of those antibodies if we had not been previously infected with SARS-CoV-2. So that's how the antibody test is specific. But you also ask some questions about the antibodies that inhibit SARS, the antibodies being used as monoclonal antibody treatments. And you are entirely correct here, because what we know in that ELISA test is that an antibody is present or absent, but we don't know if it inhibits the SARS spike protein. That would require a different type of experiment, and we aren't really sure which of those antibodies would be the best antibodies to be useful. And so we do need to think of things other than that ELISA test that will help us understand which antibodies may be useful, like in neutralization or something like that. And so your final question of, are we able to assess whether an individual has had SARS-CoV-2 based on the antibody proteins amplified after infection is completely correct.
If you have antibodies to SARS-CoV-2, that indicates that you've been infected. If you don't, it doesn't mean that you haven't been infected.
That's a good point. That's very true.
That's becoming more and more clear. I would also point out that so when the ELISAs are developed, I'm sure that the antigens that are used, the SARS-CoV-2 bits that are used, are screened for. There are things that are highly specific for SARS-CoV-2. They're not going to give a cross reaction with antibodies that are not specific for SARS-CoV-2. So we know in developing the assays that it's highly specific. I would also say, and I could probably use some insight from you on this, Brianne, that when you mount an antibody response, first of all, not everybody's antibody response is going to be the same, right?
That's correct. Yep.
Because it's sort of an individual thing. You fabricate these things in response to the infection. It's a fascinating process, and you don't necessarily just make one antibody. You make a family of antibodies that will react with different parts of the protein, and different people have different families of antibodies, and they work together to some extent, and not all of them work the same, but there are techniques that can be used to pick out of an individual cells that make each kind of antibody that you make. And that's what we talk about when we talk about monoclonal antibodies.
Exactly.
And so it becomes a whole study to figure out which monoclonal antibodies react with which part of the protein, which of them are good at neutralizing the virus, or how they work at all. They may block infection, they may kill it. There's lots of different mechanisms. So
Exactly. So usually when we do that ELISA test, we're using a whole protein or a whole virus to sort of deal with the problems that Rich just mentioned, because that way we're not asking the antibodies to bind to just one small piece of a protein. Any antibody that binds to any part of that protein will be just fine in making the assay positive.
And you could have a positive test because you have half a dozen different antibodies that react with the antigen that's on the plate.
Sure.
But don't forget those antibodies made by a bunch of different patients that seem to be all the same high affinity ones, right?
I know. That was so interesting. How similar those antibodies were from all of the different patients.
That was cool.
Yeah, it was.
I'm sure we'll hear more about that. All right.
Maybe I'll be reborn an immunologist, okay? Because it's really interesting.
It really is. Rich, you can come take my class if you want.
I'm done.
Just look at it. It's on YouTube.
It is.
Okay.
Because right now you can't go there.
Yes, true.
Brianne, can you take the next one?
Sure. Dean writes, I am a non-science trained lay listener who loves your show. For sure, there is lots I don't understand, but it still seems very informative. I am a very avid road rider, but since the pandemic, I have not gone on any group rides. My riding club is starting up again. Normally, we ride wheel to wheel, but the new guidelines require increasing the distance to 6 feet with a max number of riders of 10. I am worried that even at 6 feet, we are riding in each other's slipstream, and we just don't know where droplets will be by the time we hit them. Am I being too cautious? Thanks, Dean.
So where does bike riding fall on your chart there?
I'm looking right now. I'm not sure it's here.
Boy, that's a tough one.
All I can say is I got exercising outdoors, but that doesn't really cover it. That's a 2. It's a good question. Put it that way. When a scientist says good question, it means they don't know the answer.
Yeah. I mean, it's good that you're outside, and so you have a lot of airflow. But you're exactly right that being in each other's slipstream, I guess that's what you call it. That's what you call it, would be the issue here.
That's what they draft on each other, right?
Exactly. Yeah.
So you're clearly, I mean, I don't know how practical it is to wear a mask because you're probably breathing really hard, right?
Mm-hmm.
I think that would help, but I suspect Dean is young, and so again, low risk for disease, but you could spread it to others if you get it from someone else. So
this is not a zero risk situation.
No.
Okay?
No,
anytime you put enough people together breathing hard, yes. You're asking for trouble.
We're better at the epitope questions.
Yeah. Those I can handle.
Well,
I know how to do those experiments a little better.
We have data, right? That's the problem.
Yeah.
There's no data for bike riding and risk, so you have to speculate. And some of you don't like that we're doing that. So would you like to have someone else who has no virological expertise speculating? No?
There's too much of that going on.
If we were talking about bacteria instead of viruses, then I could ride my bike behind you with a plate and maybe try to get some data.
That's right. Well, we could put some phage in your nose and have you ride a bike, and I would hold the plate up.
Yeah,
we could do a plaque.
exactly.
Well, maybe we'll get some mail on this one.
That's the thing. You could do transmission experiments with phages, right? Because they're safe to do. But I don't know how you would start them in the bike rider in front of you. I guess you could paint them on the nares, right? And then see if they would go to the person behind. But it's not really the same because it's supposed to be coming out of the nasopharynx,
Right, exactly.
All right, one more. John writes, "Hi, ladies and gents. I work in IT. In IT, we have reverse engineering. Can COVID-19 be reverse engineered?" I like this question. The question, however, correctly stated is, can SARS-CoV-2 be reverse engineered, John? Because COVID-19 is the disease, and we can't reverse engineer a disease, but we can. We can take the viral genome. We can make a DNA copy of it. And from that, we can make more virus, or we can modify the DNA copy and make virus. And that's what I assume you mean by reverse engineering, right? We can make the virus from the genome sequence. I guess, a reverse engineered program, you get the code and you can make I don't know. I'm not sure what you mean in IT by reverse engineering, right?
So you could even, knowing the nucleotide sequence of the genome, just written on a piece of paper,
make the virus.
Chemically synthesize the DNA.
So reverse engineering is the reproduction of a product by looking at its construction or composition, right? So we can't just look at SARS-CoV-2, but as Rich says, we have the genome. We can look at it and not just look at it, actually. We can put it in. We put it in cell. We can make the genome, so we can get the virus out of cells. We can sequence it, then we can make the genome chemically and put it in cells, and we'll get the virus back. I'm not sure that's quite the same, but so I think don't you guys agree? The answer is yes. We can reverse.
Oh yeah,
I think so. Yes,
but it's a little bit different from saying, I take this pen and look at it, and I can make one by looking at how it's constructed, right?
Right.
But the virus, we're cheating because we have the genetic code. So
yes,
I want to emphasize that doesn't mean that this virus was constructed in a lab in Wuhan.
No, not at all.
Not at all. And the other thing is, we would take that genetic code and put it into a cell. We couldn't just take that genetic code and sort of make all the proteins ourselves and then build them all together. We still need a cell to help us with this process.
I like the question, though. It's good. I like it's kind of out of the box, right? All right. That is TWiV 631, microbe.tv/TWiV for the full text of letters and all the show notes. Send your questions and comments to TWiV at microbe.tv if you like what we do. We'd love your financial support, microbe.tv/contribute. You can give a little bit of money every month, a cup of coffee a month. It helps our expenses. Yes, thank you very much. And those of you, by the way, who already support us and who have joined supporting us, we really appreciate your support. It's a validation of what we are doing here. Brienne Barker is over at Drew University and on Twitter, BioProf Barker. Thanks, Brienne.
Thanks. It was great to be here.
Rich Conda is an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough, this made my day. Wonderful. Thank you.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.